Okay, welcome to this video tutorial of lipid digestion and absorption. Okay, so lipid digestion is quite a complex digestion. So it really differs from carbohydrates and protein digestion. What really differentiates these is that when we look at protein digestion, there's just direct use of those enzymes. Okay. And for the carbohydrate digestion, there are just specific enzymes which break down it. But for lipid digestion, since lipid is made up of both um, hydrophobic heads as well as um, hydrophobic hydrophilic head as well as hydrophobic tail. So it is hard for an enzyme to act on it direct, which is the lipase. Okay, so let's just break down the concept of lipid digestion. Okay, so I have this diagram here. The left side is showing the digestion of lipid, and this triangle here, whatever is here, we are going to look at that. If it get absorbed, how is it going out to, if it get to be broken down, how is it going out to be absorbed? Okay, so the digestion of lipids actually starts in the mouth. Okay, so before we start the digestion of lipids, we need to know the sources of lipids, of course. Possible sources of lipid, we have the meat fat. Okay, then we also have the dairy products like milk, cheese, all those are sources of lipid as well as the fats. Now, their digestion, mainly, they have the major digestion as well as the minor digestion. So, it just entirely depend with what book mainly you are reading, but mostly there is minimal digestion of fats or lipids in the mouth. Okay, but in the mouth there are enzymes, what we call the lipase, which is there. It just helps in it just helps in a small proportion digestion of the lipids, not as compared to the way other enzymes act on it. Okay, so this enzyme, the lipase, is actually weak and mainly it's active mostly in children, okay, mostly in infants. So the lipid digestion mainly is really happened by emulsification as well as the lipase action. So we must know to say that in what forms is this lipid going to be, um, or what are the simplest molecules that form this same lipid? Okay. So we have the triglycerides and these the molecule or chain of glycine. Okay. So under normal circumstance, at the end of breakdown of lipid, we must have triglycerides, we must have monoglycerides and the free fatty acid. Okay. So as I mentioned to say that lipid digestion is quite complex. Now, what really happens is it's like you having cooking oil on your hands, then you want to go um clean your hands just by water without anything. It's hard. Okay. So there comes in what we call biosalt. Biosalt is more like soap that we use to scrub our hands so that we the dirt can be completely removed. So the in this case, the soap is the one which is acting as the biosalt. So before lipase enzyme can act on lipids, we need biosalt to act on these bigger, larger molecules of lipid. The process of emulsification does not involve anything apart from breaking down these lipids into more like smaller units to create more area for the lipase action. So the emulsification, what really happens is that this biosalt is going to break down these bigger molecules of lipid by the process of emulsification. Now that This um, biosort has created rooms and surface area for lipase to act on them. Then the lipase action becomes so simple. So what property of biosort is enables lipid digestion? So biosort is amphipathic. It has both water loving as well as protein loving. So those areas which are protein loving, they combine with the lipids, and those areas of lipids which are water loving, they combine with water. Then that process we call it emulsification. We've broken down it smaller component. Then now this enzyme called lipase can now act on the lipid. It is important to take, there is a note that you must note to say that without uh without biosort, the the breakdown or digestion of lipids is almost impossible. Okay, so I've talked about the emulsification process, which is very much important. Now lipase can act on these emulsified fats. Okay, so lipase now can further act on these same triglycerides to form the monoglycerides and the free fatty acids. Now we've broken down these lipids into individual or smaller units. They are now ready to be absorbed. But these lipids, as always, they always have issues traveling in the in the GI, okay, because the rumen is mainly found with water, okay, and these lipids have areas which are water hating and water loving. So this biosort does not neglect, does not only break down it. So what it does is, together with protein, they try to find bonds and put these free fatty acid monoglycerides and triglycerides. They more like incorporate them together to form what we call the micelles. So the structure of a micelle is that biosort is outside, more like acting as a shield for triglyceride, monoglyceride, and free fatty acid and the glyceride, which can now be transported to the mucosa, which is the intestinal mucosa of the jejunum, so that it can be absorbed. How is it going to enable it? Is this biosort has now combined together with these component fatty acid, cholesterol, and monocyte to form the micelle. Now the micelle now can move in a furry motion, more like a boat-like, then transport this into the intestinal mucosa. Once it go into the intestinal mucosa, what happens there is it is now ready for absorption. Now, how does the lipid digestion occur? Now let's imagine this micelle has been formed and it is the one which is having all these components. Okay, so it is having the fatty acid, the cholesterol, and monoglycerides. It has now reached the jejunum area and it need to be absorbed going into the enterocyte. Okay, and it need to cross the apical membrane. Immediately after the apical membrane, it's the enterocyte. Then after the enterocyte, we have what we call the basolateral membrane. Then from the basolateral membrane, we go into the interstitial fluid. Then from the interstitial cells, we go into the capillaries, where now this should be transported into the bloodstream. Now, what happens is that this micelle does not get to be absorbed as well. So this biosalt and other components like protein which were formed does not get to be transported into the enterocyte. What happens is that biosalt and protein detaches themselves, only allowing fatty acid, cholesterol, and monoglycerides to enter inside by, the process of carrier-mediated mechanism of transport, carrier-mediated. So of course there are some small lipids like phospholipids, smaller molecules, can easily diffuse from the intestinal lumen of the jejunum going into the enterocyte. So once these components now have reached inside, these are individual components. That's why lipid digestion and absorption is really fun, because these molecules this molecule was big enough, then it need to be broken down into smaller molecules so that it can be transported. But once it is in the enterocyte, the Golgi apparatus try to package them into chylomicrons. Chylomicrons are actually bigger molecules of lipids. These triglycerides are get to be re-esterified, more like again be combined, then they form chylomicrons. Now these chylomicrons are actually bigger. They cannot bypass the basolateral membrane going into the interstitial cells. So how are they going to move from the enterocyte going into the interstitial cells? They are going to use the process of exocytosis. So exocytosis is just happens on the membrane where the vesicle just more like eat these individual component and send them inside the bypassing the interstitium. Okay. Now these chylomicrons are too large to go into the capillaries because capillaries are too small. How is it going to happen? Okay. So the absorption of lipid does not just involve movement of substance from the enterocyte and going into the blood capillaries. No. First, they use what we call the lymphatic system. So these chylomicrons actually get to be absorbed by lymphatic system. So they go into the lacteals, okay, lacteal. Then from the lacteal, they now get to be transported via the lymphatic system. Then they go into the thoracic duct. Okay, thoracic duct. Then the thoracic duct that's now going into the vena cava. Okay, so from the vena cava, so for lipid digestion, for it to go into the bloodstream, it is actually via the venous return. So it goes into the thoracic duct, then eventually from the thoracic duct, then it goes into the bloodstream now, so that it can be utilized in the bloodstream. So this is all about digestion of lipid as well as the absorption of lipid. I hope it was helpful, and don't forget to subscribe and like the video.